So the question was, can this virus start to spread from human to human? Because if it did, one can imagine that would be a pretty dangerous pandemic. Some researchers decided to ask the question experimentally. Can we make H5N1 influenza transmissible? The concern here was, is this a biosecurity risk? So might someone misuse this information?
How to Science, a podcast from the College of Literature, Science, and the Arts at the University of Michigan, Season 2. With podcast host, LSA Professor Monica Duz. This is me. The theme of the season is How to Science in Society. Season 2 explores the lives of scientists. We're interested in figuring out who are scientists, but also what it's like to do science in the context of a bigger society. After this story, I'll be back with a Q&A with the guest to talk more about how to science in society. Take a look through someone else's safety goggles.
My name is Michael Imperiale. I am a professor of microbiology and immunology at the University of Michigan. And the story I'd like to tell you about today is concerning controversies relating to research with infectious agents that is dangerous. And so lapses in biosafety in various laboratories came to light between 2012 and 2014. For example, a vial of variola virus, which is the causative agent of smallpox, was found in a closet at the National Institutes of Health. That vial of smallpox should not have been there. After smallpox was declared eradicated, all of the existing stocks of smallpox were supposed to have been destroyed, except for two places, the CDC in Atlanta and a lab in Russia, that were permitted to keep stocks of smallpox. And so that was really a biosecurity breach. The biosafety lapses were more just carelessness, where people weren't following proper protocol. The CDC accidentally shipped some H5N1 virus to a laboratory when it was supposed to be shipping H1N1, which is much less pathogenic. So this then raised people's concerns that if a lot of laboratories make viruses more pathogenic, if those viruses were to accidentally escape the lab, then that could lead to a very dangerous pandemic. I first became interested in this when I first learned about a concept that's called the dual-use dilemma. Dual use was a term that was really used in the military, and it was used to describe technologies that have both a military use and a civilian use. Okay. Work that has beneficial applications, but the results of which may also be misused. So it's a much broader definition. The dilemma then is, should we be putting restrictions on that beneficial work because we're concerned about potential misuse? There's always the potential that someone may take those same technologies and those same pieces of information and try to misuse those to cause harm. I was appointed to a national advisory board that's called the National Science Advisory Board for Biosecurity by the government to advise them on these dual-use issues. And things really came to a head in the fall of 2011. And what happened there was that a highly pathogenic avian influenza strain called H5N1. This strain was starting to arise in poultry in China and Southeast Asia. And what was frightening was that when humans were getting infected with this strain of the virus, the mortality rate was very high. It was about 60%. To put that in perspective, the 1918 influenza pandemic was the worst that we know about. The mortality rate was about 2%. When we have seasonal influenza outbreaks every year, the mortality rate of those is probably about 0.1%. The way that influenza viruses spread is through a respiratory route. So if I'm infected and I sneeze, then that puts particles out into the air that the next person can inhale and then become infected with the virus. What was happening with the H5N1 was that humans were coming in direct contact with infected birds, and that's how they were getting infected. But there was no evidence at that time that those humans were then spreading it to other humans. So the question was, can this virus start to spread from human to human? Because if it did, one can imagine that would be a pretty dangerous pandemic. So some researchers decided to ask the question experimentally, can we make H5N1 influenza transmissible in mammals? So obviously you can't do the experiment in humans, and the experiment was done in ferrets, which is the accepted experimental animal that most closely mimics humans with respect to influenza. The experimental setup is to infect a ferret in one cage and now take a naive ferret in another cage, put those cages close enough to each other where if that first ferret sneezes, those particles can make their way into the second cage, and that ferret can then inhale those particles and get infected. So the two papers reporting the transmissibility of the H5N1 virus were first submitted in 2011. The U.S. government learned about this, and they asked the advisory board to weigh in as to whether those papers should be published. The concern here was, is this a biosecurity risk? So might someone misuse this information? And so we decided that this information was dangerous enough that the overall results should be published, but the exact mutations that allow these viruses to be transmissible should be withheld. When we made that recommendation, there was a lot of pushback. There was pushback from the authors. There was pushback from the WHO, the World Health Organization, who felt this information was important for pandemic preparedness, and also from the journals themselves, who felt uncomfortable redacting information. The publishers were reluctant to redact any information because they felt that it was really their job to present the entire story to their readership. I think they felt like they were being put in an awkward position by the government when the government is asking them not to publish this. And I think we arrived at this redaction decision as a bit of a compromise because, again, there was enough concern that someone might take that information and run with it in a bad way that we wanted to put a little bit of a limit on what information got out there. And so the authors were asked to revise the papers, and we voted to recommend publication of both papers in their revised forms, along with comments as to why it was important to be publishing this information. A colleague of mine at Johns Hopkins, Arturo Casadoval, and I wrote a series of articles in which we tried to make the point that the whole concept of dual-use research needed to be reconsidered just because it's too fuzzy of a concept, and waiting until the work reached the publication stage was really too late in the game. You know, these types of experiments are going to become more common, and hopefully we have a little bit of a better process now in place to work with them.
This sort of reminds me of a lot of discussions people were having about nuclear power because it's cleaner energy compared to fossil fuels in general. But there's also clear risks that are potentially catastrophic. Can you draw some parallel between the two? And since we're still battling with this dual use dilemma and this controversy that we have between knowledge and potential risk, is there anything that we can learn from discussions that people have had about nuclear power and how to safely do experiments?
I think the difference with respect to the life sciences is twofold. Number one is that the resources that are necessary to develop a nuclear program, if you want to deliberately cause harm, are very, very large. It's very expensive, requires very, very high levels of expertise. In the life sciences, the resources that are necessary to make an influenza virus are not that high. The expertise is not at that same level. You can imagine someone or some group or some nation state that wants to cause harm, reconstructing that virus in the laboratory. So the technology for producing an influenza virus is pretty straightforward. It doesn't require highly sophisticated laboratory skills. It does require some expertise, but it's not rocket science, I guess you could say. And it's easier to hide. So someone can do this in their garage or their basement. You know, you can't develop a nuclear weapon in your garage or your basement. So I think that's one difference. And then the other difference is that I think just humans have more of sort of an inherent or innate fear of infectious agents.
in light of what you just said, how do we do these experiments or this quote unquote dangerous science in a safe way?
I think if it is, for example, an infectious agent, that we're going to have the infrastructure in place that will, number one, prevent it from accidentally getting out. And number two, if we really think it's a biosecurity risk, to have those sorts of physical security, locks, those sorts of things. Number two, we have to make sure that the individuals who are doing the work are properly trained, because that may be the weakest link is human error. The other thing I think we have to remember is that oftentimes when we're doing experiments in the laboratory, we get unexpected results, and we may unexpectedly isolate something that has a gain of function, right? And so I think we have to be aware of that possibility and then know what we're going to do when that happens, right? That we need to stop and think, are we going to continue these experiments? How are we going to publish the results? Who do we need to talk to get an informed opinion about the way to move forward?
In terms of the board, there were 2 members of the public, and they represent all of us as public. So what was their take on how much transparency there should be in public research?
So I should talk a little bit about the composition of the Biosecurity Board. So there were scientists, there were individuals who had background in science policy, lawyers. There are 2 members of the public on the board who are not working on these things day in and day out, bring a different perspective, right? And so the expertise on the board was quite diverse. I think we all realized that we had a common goal here, which was to do what's best. So scientific research needs to be transparent because, number one, that allows other laboratories to verify and reproduce the work that was done. And secondly, that work will act as a foundation for further work. In this particular case, the argument for publishing the work rested on a few things. Number one was that, as I mentioned earlier, it was just important to know, is it possible that this virus could become transmissible? Some pathogens are highly contagious. Measles is probably the best example of that. Other pathogens are not very contagious. In the case of the avian H5N1, the strains that are circulating in birds cannot be transmitted from one human to another without heroic efforts. The second thing then is, what can we learn about what the determinants are of transmissibility? Because that then may allow us to do better surveillance when these viruses are infecting poultry or other birds. And secondly, it also allows us to ask questions about whether or how to best approach developing vaccines against this particular strain. The public is paying for this research, and the public is the beneficiary of the research. And so I agree with that we have an obligation to the public to ensure that we are doing what's right as well as what's best.
I guess what I'm hearing from you is that in many ways, we still haven't really resolved this dual-use dilemma.
I think in many ways we have not. I think biotechnology is rapidly advancing. It's hard to keep up policy-wise, oversight-wise, with all the new advances. The board continues to meet. I know it's still discussing these types of issues, and hopefully it's going to continue to provide recommendations to the U.S. government on how to proceed here. Having said that, it is an advisory board, and the government could have decided to ignore our advice. Fortunately, they did not.
What about joining our Facebook How to Science podcast group, where you can learn more about science, about how to science, and maybe get to talk to scientists about science news? Join the How to Science Facebook community, where we want to pop the bubble of the lab and see what it's like to do science in society. Make sure you subscribe. It's a hard word, subscribe. Okay. Subscribe to the podcast. Help your friends find out how to science by giving us a 5-star rating on iTunes. And I honestly can barely pronounce the word algorithm, but something about the iTunes algorithm really helps other people that might never otherwise hear about it find it and listen to it. How to science. How to science. How to Science.
The How to Science podcast is recorded at WCBN FM, the student-run, community-supported radio station on the campus of the University of Michigan.
I like risotto, actually. My favorite pasta is probably just like broccolini pasta, like with broccoli rabe.